And this way, you will have had an opportunity to at least think about gram staining prior to being tested on certain aspects of gram staining. And then, when you actually go into the lab and you do the gram stain, you'll have been exposed to it a number of times. And therefore, with luck, that will mean that you have some familiarity with it before you actually set out to do it. Okay, so the gram stain. So why do we gram stain? We gram stain because we want to distinguish between cells that have thick cell walls, we'll get to what a cell wall is, and no membrane surrounding those cell walls, and cells that have thin cell walls and have a membrane surrounding those cell walls. We also have cells that have no cell walls, the mycoplasma, and we also have cells that have interesting things in addition to the cell wall and the membranes that are associated with it. And it's confusing because it's the mycobacterium especially that have these other things, these mycolic acids, these waxy substances. So mycoplasma lacks cell walls, and mycobacterium has something in addition to just the cell wall and the membranes. But we're not going to worry about mycoplasma right now, which would stain gram-negative, I should note. And we're not going to worry about the mycobacterium, because that's something that we would look at if we were using the acid-fast stain, which is something we'll get to in lab, but it's not something we're going to concern ourselves with right now. Instead, what we're going to talk about is gram-positive and gram-negative. Gram was a person, so we capitalize gram. It is not a reference to a unit of weight. And so we have gram-positive, and we have gram, oops, there we go, negative. Or you could say gram positive and gram negative. So the gram positives are positive because they stain with what we're using in the gram stain. And they stain that way because they've got thick cell walls and they're lacking this, what we call the outer membrane, the membrane that surrounds the cell wall. So thick, not think, thick cell wall. Whereas the gram negatives have a thin cell wall, easily disrupted. And no outer membrane, and outer membrane. Or yes, outer membrane. Easily disrupted, difficult to disrupt, or more precise, more difficult to disrupt, easier to disrupt. And disrupting of the cell envelope consists of a combination of the cell wall and the membranes associated with it. The disruption of these cell walls is what is the basis of gram staining. If we can disrupt the cell walls, then we can remove the stain, and as a consequence it stains, that's not a contradiction, we'll get to why, on gram negative. And if we fail to remove the cell envelope, then we don't remove the stain, and therefore the stain remains and we call it gram positive. The process of gram staining involves making something that is big and difficult to pull out of a cell without disrupting the cell envelope. And this big thing is made by combining the stain crystal violet with iodine. The iodine is also called Graham's iodine. And it's also called mordant. And what happens is that you first, like you were simple staining, stain with the crystal violet, and then you wash off the excess crystal violet using water. Remember, you've put down your smear, you've fixed your smear. You've done so without an unbelievable amount of cells. Because if you do have an unbelievable amount of cells in your smear when you're gram staining, it's going to be very difficult to distinguish between the gram positives and the gram negatives, because the step where you're trying to see whether you can disrupt the cell envelopes, you're going to have so many bacteria there that it's going to be difficult to figure out how to add enough of this, what's known as decolorizing agent, to remove or to destroy the cell wall sufficiently that the stain comes out. So you don't want to have a huge number of cells. What is a huge number of cells? A huge number of cells means the cells are piled up on top of each other. What you want are cells that are reasonably well separated from one another. They don't have to be hugely separated from one another, but a nice little layer that doesn't consist of just cell, 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 cell is what you're shooting for when you're making your smear. Of course, you fixed your smear, you passed it over the flame. Then you added the crystal violet, and now the cells are stained with crystal violet, which is fine. And then you wash it, water, and you then flood with grams iodine. Basically, it's like you've added another stain. Only in this case, what happens is the crystal violet interacts with the iodine, and it creates something a little different. And this something that is a little different is difficult to remove using the decolorizing agent. So the decolorizing agent basically is an organic solvent. So you've added the crystal violet, you've added the grams iodine, let it sit. It forms this somewhat insoluble agent that you have to disrupt the cell wall, cell envelope in order to remove. Then you wash it again with water. And then you decolorize. So the decolorizing agent can be acetone, alcohol, it can be simply alcohol. It's something that will dissolve the crystal violet. And it is something that will dissolve the outer membrane. And you add this decolorizing agent, this is where it becomes an art. You add this decolorizing agent until you stop removing color. So you have to add it drop by drop by drop until the color is no longer running off of your smear. Now this is going to be relatively easy if you don't have tons and tons of cells making up your smear. If you do have tons and tons of cells making up your smear, it's not going to be very easy, and it's not going to work out as well for you. But once you have decolorized, then what you do is you wash again with water, and you add a counterstain. Now the counterstain is the reason why saying that something stains gram-negative is not a contradiction. The gram-negative cells do actually end up being stained in the end. They're just not stained gram-positive. They're not stained with this combination of crystal violet and iodine. And this counterstain is called saffron. Hopefully that's spelled reasonably well. So this combination of crystal violet and then mordant will bestow a purple color to the cells, and the saffronin will bestow a pink color to the cells. So if you, after you've decolorized, if you haven't removed the cell envelope and thereby this combination of crystal violet and iodine, the cells will be purple. When you add the saffronin will not really change that color of purple. And so the cells will be purple, and therefore they will be gram-positive. Or we take away from that in fact we have gram-positive cells. Whether or not they're gram-positive is highly dependent upon your destaining step, your decolorizing step. And that's because if you decolorize too much, you're going to turn your gram-positive cells into gram-negative cells. If the cells are too old, they're going to be too delicate, and you're going to turn your gram-positive cells into gram-negative cells. There's a real art to doing this gram staining. On the other hand, if you have removed the purple color, when you add the saffronin, you will now have pink cells, and you can at least see your cells. You can see that they're there. And this pink color is an indication of gram-negative. So pink gram-negative, so for example, E. coli. Purple gram-positive, for example, Staphylococcus aureus. We've got a bunch of different types of bacteria that we categorize as gram-positive, a bunch of different types that we categorize as gram-negative. There's a fundamental difference between the gram-positives and gram-negatives. The gram-positives have these thick cell walls and no outer membrane. The gram-negatives have a thin cell wall and an outer membrane. The cell envelopes of the gram-negatives are more complicated than those of the gram-positives because of that outer membrane. Because they basically, cell walls consist of a sandwich. Outer membrane, cell wall, and then an inner membrane, which is the same thing as a plasma membrane, which is the same thing as a cytoplasmic membrane. On the other hand, the gram-positives have these big thick cell walls, but no outer membrane, and then they've got a plasma membrane sitting underneath the cell wall. So it goes cytoplasm, plasma membrane, cell wall, away from the cell. There's even more associated with the cells than necessarily just their cell envelope, but we'll stop there in talking about the anatomy. So there's a fundamental difference. And you've got some pathogens getting to medical microbiology that are gram-positives. For example, among pathogens that are gram-positives, Staphylococcus aureus, which I already mentioned, Bacillus anthracis. So all the genus Bacillus are gram-positives. The genus that gives rise to, for example, tetanus and botulism, as well as gas gangrene, which is the Clostridia, genus Clostridium. They're gram-positive as well. The gram-negatives, on the other hand, is a whole slew of gram-negatives. So Escherichia coli, it can be a pathogen. Salmonella, gram-negative. Yersinia, which causes plague, is a gram-negative. And so on and so forth. All the spirochetes, such as the cause of syphilis, Treponema pallidum, are gram-negatives. Lots and lots and lots of gram-negatives out there. Among the pathogens, although they can be pretty nasty, relatively few are gram-positive. And then there's lots and lots of gram-negatives. So gram staining, the process of gram staining. So we make our smear, and then we fix it, and then we flood with crystal violet. We then wash off the crystal violet. We flood with grams iodine. Pour off the crystal violet. We pour off the grams iodine, and then we wash that, and then we add the decolorizing agent. And we have to decolorize the right amount of time, which is a real art. And then we wash that again with water. We add saffron in as our counterstain, and the counterstain then dyes these cells pink, or stains these cells pink, if in fact we have washed away the purple coloration. So gram staining.